All right, so this is kind of going to add on to the previous video. We talked about resting membrane potential, and we're going to talk about what are called local potential changes in this one. So just a refresher, we talked about the resting membrane potential that is set up. It was, remember, negative 70 millivolts, and it is set up by the difference of the potassium on the inside and the sodium on the outside. And so what we're going to talk about is a disruption of that resting membrane potential in a certain particular area. Now, on the last one, I did briefly mention that there's a distinct dividing line. We called this area the axonal hillock. You should have looked at that a little bit in lab, or are going to. It's a specific and special area where the axon is coming off the cell body, and it is a dividing line. Everything that's on this side, those little ion channels that we talked about, they're going to be electrically controlled over here. The ones that are on this side are going to have various different things. Sometimes it's pressure, sometimes it's chemicals that are going to open them. And all the things on this side that are going to be changed are going to be called local potential changes. And that's what we look at in this little video. So local potential changes are going to be disruptions in the resting membrane potential. So when this says it detects stimuli, so when something happens, and again, I wish I kind of had a picture here. It's going to be a bad picture, but just bear with me. So I've got that's my axon. I've got these things over here that are dendrites, sticking out. There's a dividing line here. And so let's say this is going out to my fingernail, right? That's supposed to be my thumbnail, we'll say. And I touch something that's kind of hot. I'm going to have a thermoreceptor here, and it's going to have, that would be considered stimuli, and it's going to open the receptor things, and it's going to allow for sodium to come in, and it's going to end up causing a little wave of electrical impulses going back to this cell body. That is called a local potential change. And so we also have where other neurons are going to come in. So let's say this is the axon of another neuron that's wanting to talk to it. It's going to drop off neurotransmitters here, and they're going to cause a change in the resting membrane potential at that area. But it is a local potential change. We're going to look at in another video how everything that happens here is called an action potential. Now, these local potential changes, which we're saying is just a changing in the resting membrane potential, is going to be caused by the opening, as it says here, of gated ion channels. And as that happens, the membrane potential is going to become more negative. If, I should say, it becomes more negative, it is called hyperpolarized. So if I have 0 at the scale here, and I have negative 70 here, and let's say it goes down to negative 80, right? It has been hyperpolarized. So this will say that this little dividing line here is, that's where normal is, and it goes down. That's a hyperpolarized axon. If that goes closer to 0 or becomes less negative, that is called depolarization. Less negative means, which is another way of saying more positive, is a depolarization. And so I've got all of these local potential changes coming in. And now neuron cell bodies, like in your cranial cavity, the central nervous system, your brain, the neurons in your brain, each individual neuron is going to have tens of thousands of things attached to it that are giving it information. So it's got a lot of local potential changes. So as these local potential changes all accumulate inside this cell body here, they're going to kind of build up and kind of work their way down to the axonal hillock, that little spot where the axon's coming off. And they get graded together, and that's why sometimes they can be called a graded potential change. Graded stimulus. And so there's going to be some stimulus that's going to push things closer to zero, which they are called depolarizing stimuluses, or I'm going to have some things that push things farther away from zero, make it more negative, and those are hyperpolarizing. And so as these all work together, if those local potential changes, and again, I'm going to try to erase some of this here. If those local potential changes, and I'm going to get that scribbly stuff out, can add up to what is negative 55 millivolts. So let me draw this again. I shouldn't have erased everything. So we're saying this is negative 70. This is 0. All right, and so this is negative 80. All right, so if it gets farther a negative, that is hyperpolarized. If it gets closer to 0, it is called depolarized. And right about here, there's a magic number called negative 55. And negative 55 is called threshold, because as all of those local changes kind of work their way together towards that axonal hillock, if they are added together and produce a stimulus that reaches negative 55, then we will have an action potential created, and an impulse will go down the axon to wherever the terminal is. And if it doesn't add up to reach negative 55, nothing happens. It's either going to or it won't. And so you've got, let's just say I've got one neuron here. Again, I'll draw up kind of a bad version of it. And I've got one neuron here. This is where the axonal hillock is. And I've got 3 different local potential changes come in. I'm going to say one of them is positive 10 millivolts. One of them is negative 10 millivolts. And the other one is positive 5 millivolts. All right, so what would happen is they would all work together and add together as they got down to the axonal hillock, and the total change would only be positive 5. Right? So I would only get to, if I started at negative 70, that means I only get to a negative 65. I've been depolarized, but it's not depolarized enough. So let's say this one, instead of 5, let's say it is 50. So now I've got a 40. Mark that out, I'm not going to erase it. So I've got a positive 40. That will definitely get me to negative 55, and then I've got an action potential. Now we're going to look at action potentials. This amount here doesn't create a strong, a more intense feeling. Once it hits negative 55, it sends a signal. If it's a huge thing, if it's positive 40 stimulus, or if it's just positive 15, which is just enough to get us to negative 55, right? So I mean, again, just to show you where I'm coming from, if I take negative 70 and I add positive 15 to it, it is going to end up being a negative 55, which is threshold, and that's going to send an action potential. So I hope that makes sense. That's why they're called that's why sometimes they're called graded potentials, because they all are lumped together. But again, in here, they're called local potential changes, and they are everything that happens in the cell body and dendrites. And there's no rhyme or reason. There's no set order. They can be really large or really small. They can be positive or negative. And so they're kind of all working together to try and reach, again, what we call this threshold potential of a negative 55 in order to create an action potential. And so on this chart, you can kind of see it. If I was to draw, so this would be where negative 50 is. So this right here would be about where negative 55 is. And if you look, I've got this little area, which is the local potentials. As it's come through, they've all graded together, and they hit this. And as soon as they hit this, they shoot straight up. And that is the action potential. Now, action potentials are what we're going to look at in another slide, in another video, but action potentials are always the same. They are going to always be the same. They're a cookie cutter, where local potentials can be positive or negative. They can be big or small. All action potentials will have the same value. It is going to be like this little electrical wave going down the axon to the synapse. So this is showing the difference. Again, this is where the axonal hillock is. All of these on this side, as it says, are voltage gated channels. All the ones on this side are either going to be chemical, or it can be pressure, or different ones. Here it says chemically gated channels, whatever. They cause disruptions, and here it kind of shows that these local potential changes did not get up to threshold, so nothing happens. This one, it shows that once those local ones got to this point, boom, it's on. We've got this quick influx of sodium ions. We're going to talk about it the next time. So negative 55 is what's called threshold. So the local potential changes have to add up to a positive 15 millivolt difference. That's how you get from negative 70 to negative 55. So again, hopefully that's making sense.